We'll start soon. I'm going to go back to the slide presentation. And here we go. You remember that we had covered the anatomy of the muscle. And the thing to remember is a tube within a tube within a tube within a tube. Smaller tubes clustered together, gathered into larger tubes. So the muscle with its epimysium contains bundles called fascicles with its perimysium, and fibers with its endomysium. Fibers are the closest thing we have to a cell in muscles. It is, in fact, a bundle of the smallest tube of all called a myofibril. Myofibrils are strings of sarcomeres, and it is the sarcomere that is the contractile unit. That is what's important. When we send a nerve impulse to a skeletal muscle and it contracts, pulling on the ends and moving our skeleton, it is actually the sarcomere that's doing that contraction. Here's a real picture of a muscle fiber. It has those stripes because the sarcomeres are lined up in register with Z line of one myofibril in exactly the same place as the Z line of the next myofibril. So it has these stripes or striations. It's a very, very long, quote, cell, end quote, one that has many nuclei because of the fusion of myoblasts that form it. At the bottom, you can see up to 30 centimeters in length. What is that, 11 inches? So it's very, very unusual as a fiber. But what makes it the closest thing we have to the single cell is that it is enclosed by the last membrane, the endomysium and sarcolemma, enclose the myofibrils in a bundle within the myofibril. And so it is a common sarcoplasm that bathes them all. The structure of the individual myofibril within the fiber is shown here, wrapped. Each sarcomere is wrapped in a net-like membrane called the Sarcoplasmic Reticulum, shown here in blue, and then tied at each end, almost as if in a bundle, like ribbon on a package, with those yellow T-tubules. That creates actually three spaces inside. The sarcoplasm bathes the entire inside of the myofibril, including the surfaces of those blue and yellow structures, and penetrating through the entire interior of the individual sarcomere. The second space is the space that's inside the blue membrane, the sarcoplasmic reticulum. This is a space where we will set up the resting muscle by storing calcium and activate the stimulation by releasing it. The T-tubule, the yellow tubes, are the third space, and this is the space that will bring the electrical nerve impulse into the depth of the fiber. This is what it looks like with the scanning electron microscope. And look at that magnification, 64,000 times magnified. And so the dark black lines, called the Z lines, show you the boundary of one sarcomere. That adjacent space that is light colored is a space that only the thin fibers called actin will go across. They are hooked to the Z line and they move toward the middle line, but do not reach it. That middle dense area called the A band is where the thick fibers or myosin fibers are concentrated. They are bonded together in that M line, but they extend toward the Z line but do not reach it. It is that area of overlap between actin and myosin, shown on the right side of this lower diagram, where actin and myosin are lying next to each other. That area of overlap is where the cross bridges will form and contraction will be produced. So this structure for the sarcomere sets us up to understand how the process works. This shows a diagram of an entire sarcomere, the two end lines, the Z plate or Z line, the middle where the thick fibers are, and that area of overlap between the purple myosin and red actin fibers. This slide on the right side shows the actin, showing how it's bonded together at the end line, the Z line, and how it has a coiled experience enlarged down below. In addition to the actin globules themselves, you have a kind of a, looks like a wire strand, the tropomyosin that holds them together, and troponin, a molecule that actually inactivates the site until a stimulation arrives. The myosin fibers are bundles of fibers, look like a golf club with a flexible head. To all students, I'm getting some feedback. Is there a question? If there's no question, please mute your microphone until the lecture is done. Okay, back to the myosin strand. You can see it's like a bundle of myosin molecules with the heads sticking out in every direction. Now, this is going to allow our understanding of how the cross grids actually form on the actin and pull them toward the M line. So here we go. The overview: a nerve impulse arriving at the membrane of the fiber, that electrical impulse moving down the T-tubules to the sarcoplasmic reticulum and keying the release of calcium. That's going to unlock the troponin lock and allow cross bridges to pull the actin toward the midline. That produces a contraction. Here is the muscle fiber. You can see the striations that represent that thick myosin middle to each of the sarcomeres arranged in register across the fiber, myofibril by myofibril. But more important, we see the where the nerve approaches the sarcolemma, you have a gap. That's called the synapse. And that synapse is shown here, synaptic cleft, space between the nerve and the muscle fiber membrane. Now that leads to a bit of terminology. The nerve impulse will be coming down that blue membrane. We call that then the presynaptic membrane. The synapse is the space itself, shown here with some additional molecules called acetylcholine esterase. And the more flesh-colored or pink membrane is the muscle fiber itself. That is the postsynaptic membrane. This is a resting situation. The nerve is loaded with vesicles full of something called a neurotransmitter substance. This is a chemical that provides the transfer of activation from the nerve to the sensitive membrane of the muscle fiber, the sarcolemma. That chemical step is absolutely necessary. And by the way, this mechanism is the same mechanism we will use to explain nerve to nerve impulse transfer. So here we are at rest, and all of a sudden, here comes an electrical impulse, a nervous impulse down the membrane. We call that nervous impulse an action potential because it is actually a measurable electric disturbance. The voltmeter would jump and the ammeter would jump as this travels across the nerve membrane. When it arrives, it causes those vesicles to fuse with the presynaptic membrane and dump their neurotransmitter. The neurotransmitter here is called ACH. That stands for acetylcholine. It is our best understood and most studied neurotransmitter. So here it comes. diffusion and dumping. The neurotransmitter then floods the synapse and binds the receptors that are on the postsynaptic membrane. That causes gates in the postsynaptic membrane to open, and as you notice, sodium rushes across. That completely changes the electrical balance. And if you'll allow me to consider when you are at rest, when you originate a nerve impulse, I'm going to call it firing the membrane. That transfer of sodium fires that membrane. See how it's turned red? And it begins emanating out in a self-perpetuating way from that point of stimulation. So, and the next slide, what you're going to see is several things. You can see the remnants of that red impulse. It has reached the opening to a T-tubule and is traveling down and penetrating the interior of the fiber. Let me also say that this action potential is traveling all over the outside of the sarcolemma for that fiber, doing the same thing at every T-tubule, activating the sarcomeres within. The other thing you see happening as the nerve impulse passes is that the acetylcholinesterase has bound those acetylcholine molecules and stripped them off the receptors, which allows those sodium gates to close and the membrane to go back to an at-rest position. I should comment that acetylcholine is then harvested and broken down. Some parts of it are thrown away. The choline piece is transported back into the nerve right there and used right there to make more acetylcholine and to repackage it in the vesicles, as you see here. That repackaging is going to prepare it for the next nerve impulse's arrival. So let's follow that impulse into the fiber. We took it to the opening of the T-tubule and down around. You can see in the upper left how the yellow T-tubule wraps around the sarcoplasmic reticulum. And on the left, that bar shows what is inside before the nerve impulse gets there. You notice the separation of the end view of those two myosin strands and their surround of that central actin strand. You'll notice the troponin on top of, or the tropomyosin on top of the yellow active site. So this is blocked. There's no way it can form a cross bridge. In the middle bar, you see the calcium arriving, and those troponin strands are rotated off by the presence of calcium. That allows the right figure where the myosin forms a cross bridge. It basically flexes down, and the hip of that myosin binds to the yellow active site of the actin. Now, this is what it looks like from the side when we go through the same sequence. Those myosin heads labeled ADP plus P are at rest and are loaded and ready to go. So in this figure, the end plate would be to the right of the figure. The midline would be to the left. And to contract, the actin has to move from right to left. The ADP plus P shows that those myosin heads are ready to go, but the troponin is blocking the active sites just as the calcium ions arrive. You'll notice the rotation from here to here as the tropomyosin troponin rotates off, revealing the active sites. That exposure allows the cross bridge to form. So watch the myosin heads as they flex down and bind and actin. So that's the first step after unlocking. You bind. The binding causes that myosin head to flex toward the midline. There. I'm going to go back. This is the power stroke, moving it 1 unit closer to the midline. So bind, flex. The next step is You notice when you flex that ADP plus P is released. That's going to go back over to a mitochondrium, and those ADPs are going to be rephosphorylated into ATP and cycled right back into the system. So bind, flex, release, and when you release, the ATP that is present binds the head. When it binds the head, it's broken down into ADP plus P, but remains bound to the head as it recovers. So let's go back and look at the 4 steps. Bind, flex, release, and reload. This is going to repeat. We've moved the actin 1 unit toward the midline, and as long as calcium is present and the stimulation continues, we're going to bind, flex, release, and reload for each of these myosin heads, pulling the actin strand in the direction of the midline of the sarcomere. Now, this is only showing 2 myosin strands. There will be 2 more on the surface that is above the surface of your screen and 2 more behind. So multiple heads will be working repeatedly all along the length of the actin, forming these cross bridges repeatedly and actively pulling the actin and shortening the sarcomere. If this myofibril that you see here is stimulated and it is not anchored to a hard structure, then basically the myofibril contracts around its center. It pulls in both ends. This is important in soft structures like the heart or other organ walls. The digestive system, the respiratory system, the urinary system, the reproductive system, all can have smooth muscle walls, and those smooth muscles contract to produce movement. On the skeletal side, we have a number of muscles that are not anchored to the skeleton to produce movement. So the Orbicularis Oris is a circular muscle in the soft tissues of the face surrounding the mouth. When the Orbicularis Oris contracts, it is like pulling the drawstring on a pair of sweatpants, and the hole simply gets smaller. When you purse your lips, make that kissy face or fish mouth, that's the contraction of a soft tissue muscle, in this case, the Orbicularis Oris. When you anchor one end to a skeletal muscle, you typically have an end that you're pulling against, shown in the B figure on the left side. So when you stimulate this myofibril, it will pull one end toward the other. So with skeletal muscles, one end typically remains fixed while the other end moves toward it. The end that remains fixed is called the origin, and the muscle that moves toward it is called the insertion. We'll get into this when we get to muscle system. There are, of course, summaries. This shows the contraction review that we just covered to explain the contraction of the muscle. When the nerve stimulation stops, then the contraction stops. But a couple of things need to happen to allow for muscle relaxation. This is when recovery happens. So the nerve stimulation is gone. So the synapse is emptied out. The calcium is transported back to the sarcoplasmic reticulum. When that happens, the tropomyosin troponin rotates back on the active site and basically locks the muscle so that it cannot form cross bridges. Contraction ends at that point, and during relaxation, some other force will stretch the muscle back out to its starting position. My favorite muscle, the biceps brachii, is one that flexes the elbow lifting the forearm and hand so that if I'm in an upright position, gravity will stretch it back out when I relax it. If I'm lying on my side and I want to extend that elbow back out, I have to use a muscle on the other side of the joint, in that case, the triceps brachii. Each sarcomere has a usable length of contraction. It does not go from maximum length down to zero, because as you can see by the central and topmost diagram here, it begins with a certain area of overlap. Going to the left, the usable tension shown from the top diagram to the upper left diagram, when the actin strands meet that midpoint, you have used up the entire length of that sarcomere. If you continue and those actin strands punch through the midpoint, then they're basically working against the direction of contraction, so the tension drops way off. Likewise, when you're stretching a sarcomere out, this is especially important when you're actively using your muscles to their maximum level, it's very possible that you could stretch the sarcomere out, as you see on the right-hand side from the right down. If you overstretch the sarcomere, as in the lower right figure, you lose the ability to have overlap between myosin and actin, and consequently you lose the ability to form cross bridges. So you can see the red line, the tension drops way off. The point of this figure in the center is to show you this normal range of flexibility, of contraction and stretching, that usable length of the sarcomere. The muscle, as you go up from the myofibril to the fiber to the fascicle to the muscle, that means that each muscle has a certain amount of movement built into it based on its size and the length and the number of sarcomeres that it has. This single contraction is called a twitch, and we'll think about how it adds up, sarcomere to myofibril to fiber to fascicle from now on. So to look at how muscles in the skeletal system actually work, are we moving something light, intermediate, or heavy? Do we want to move it quickly or slowly? What's the load and what's the duration of that contraction? It leads to different considerations for how we use our skeletal muscles. Is this an easily accomplished movement where we could basically do the movement indefinitely? Or is this something that has a high intensity of ATP use and stimulation, and therefore we won't last as long if we're trying to perform on that level? It leads to these different patterns, as you see here, of muscular contraction. Now, if you can think in terms of a myofibril and the tension it's generated, this addresses different patterns of load and duration. In A, the pattern we call Treppie, you'll notice that the green arrows, which are the nerve impulses, are spaced out, and they allow the muscle to generate tension and then relax all the way back down to zero before we stimulate it again. Obviously, this is a very low load and a long duration, so these are Treppie contractions, which basically are sustainable over indefinite periods of time. If we move those arrows closer together, as in the right diagram, we see tension building to a higher and higher level. What you should notice is that there's a period of tension building, and as it starts to relax, the next nerve impulse arrives before the muscle has completely recovered zero tension. So it builds to a higher level of tension. However, you'll notice, comparing to the left figure, it's not anywhere near the maximum tension of the muscle. And when relaxation occurs in this pattern, B, wave summation, it returns to zero quickly. However, if you continue to call for more and more muscular power, you get incomplete tetanus. If you do wave summation up to a level and sustain it below the maximum tension that's possible in the muscle. This is called incomplete tetanus because it is a long duration contraction. So, for example, you're lifting a box that's heavy and you're carrying it out to the moving truck. That's going to require this kind of incomplete tetanus contraction. The heavier the box, the higher that level of tension is maintained. Finally, complete tetanus, you notice we've crammed those areas together, and that's taken you all the way up to the limit of this muscle. That muscle is essentially as hard as if it were cramped, and it is sustained at maximum level until the nerve impulse goes away and it returns to zero tension. Another alternative to that is that incomplete tetanus, you basically use up all your oxygen and all your ATP pretty quickly. And as a result, the muscle will start to lose tension in a short period of time. There are a couple of mechanisms built into the body to ensure longer duration and better operation. And this concept is a very wonderful concept that uses the nervous system to distribute the muscular contraction among different muscle fibers. And they're shown here in groups. There are 3 of them, blue, red, and purple, arranged as fibers. In this case, we are looking at a specific fascicle. The important thing to notice is that from the motor control area of the spinal cord, that means there are 3 different nerves attaching into this muscle fascicle. They can then sequence their firing so that they are not all at the same time, but that they alternate among red, blue, and purple. And that produces a contraction pattern like, that produces a contraction pattern that will allow the purple group to fire, relax, and return completely to zero. When it's halfway relaxed, the blue group fires, and when the blue group is halfway relaxed, the red group fires. So taken together, these 3 motor units are working in sequence, overlapping, generating both a higher level of tension than any of them could do alone, but also allowing for greater endurance since each is essentially individually showing a Treppie pattern. So what does that mean in terms of energy consumption? Within the muscle, we have 3 sources of energy. There is some ATP at all times, a very small amount. And look at that top line, 10 twitches for a typical muscle fiber, which will last about 2 seconds. So if you are confronted with a high level of activity, the ATP that's there is gone in 2 seconds. The creatinine phosphate is a storage device which basically holds phosphate, but basically releases ATP when called upon. If you have an emergency situation, another 70 twitches or another 15 seconds of energy that do not depend on your oxygen is available. But that basically represents, the ATP and the CP represents your emergency startup, and it lasts basically 17 seconds. At that point, the glycogen that's stored in your muscle will undergo aerobic respiration, beginning with glycolysis, which is anaerobic, and then the breakdown to CO2 and water, producing ATP in the mitochondrion. There's about 100 millimoles of glycogen in that fiber, and if it will be limited to a maximum based on the amount of oxygen present, but you'll notice that the anaerobic gives you another 2 minutes of activity. And if you can slow down to the point that you're just using aerobic, another 40 minutes of endurance. So the body is switching off using these different sources, and it might be useful to go through and look at different levels of activity in the muscle fiber. So that's where we're starting. This is at rest. What's happening in your body? Well, the blood vessels are delivering fatty acids and glucose. They're also delivering amino acids, but basically those are different carbon fragments, and they would act pretty much like the glucose. What your body is doing is using fatty acids principally in your mitochondria to take care of all your ATP needs. Now, the oxygen demands are slow, and so you're breathing at rest normally, and the CO2 production is relatively low. The ATP that is produced is being used to support your bodily activity of all organ systems, and the extra ATP is charging up that creatine to creatine phosphate. The glucose that arrives is being stored in the muscle as glycogen until those, if you'll allow me the idea of a tank, there's a certain space for glycogen in the muscle. And when that space is full, then that glucose would go on to other purposes. So at rest, there's low oxygen consumption, easy breathing, low CO2 production. Recharge your creatine phosphate and glycogen tanks, and then the nutrients are going on to other storage. Perhaps they're stored in the liver, perhaps they're stored as fat. But then you have a chance to work out. And that activity I'm going to call moderate, right up to the level of the limits of oxygen consumption. Fatty acids continue to provide a background amount of ATP by feeding directly in through the mitochondria. Now, it turns out this is not through the glycolysis process, it is through a thing called beta oxidation, but it does produce a lot of ATP. Because it's not an emergency situation, the creatine phosphate remains stored and it is not shown here. The glycogen is broken back down into glucose, shows the production of pyruvate by glycolysis, and then the 3 carbon fragments are fed into the mitochondrion, where they will be broken down to CO2 and water. So it is mainly fatty acids and carbohydrate that is activated during moderate activity that is up to your oxygen limits. So let's say you're a runner. Let's say you're running 45 miles a week in order to condition. It is this pattern that shows you your daily flux of energy molecules to produce ATP in the muscles. That would also mean that you go out on a 6-mile run as a workout on Wednesday, and it depletes all of these tanks. That means that part of your recovery and rest is to refill the glycogen tank. During this activity, you would be breathing harder, your oxygen consumption and CO2 production would go way up. But above that, you still actually have an emergency reserve. I was interested when I ran my marathons. After running 26 miles, I had 385 yards to go. And I was tired and beginning to become sore. But I found out if I tried to sprint, I had a small reserve of extra energy that allowed me to increase my speed. Now, that was for a very short time. But that's the emergency reserve that adds on to the top at moderate activity, what we call sympathetic stimulation. Hormones will improve the reception at the synapse and nerve transfer. And in addition to the glycogen and the supply of fatty acids. By the way, that background stuff is still going on. But what happens is that we begin to use glucose in a different way. Whereas we can only produce CO2 and water at the level of the supply of oxygen. Above that, we can continue to break glucose down into pyruvate, the process called glycolysis, and harvest a few extra ATP. You'll notice that as the glycolysis kicked in, that creatine phosphate is also yielding extra ATP. So those two ATPs added on top of the moderate activity is what will give you the ability to do that short sprint. Now, that doesn't last very long. And the extra pyruvate in our body is harvested and stacked up as lactic acid. This is what causes your overworked muscles to bulge. It also explains when you work in a very concerted way why the next day or the day after you wake up and you're sore. It's from the lactic acid in the muscles, and it's actually a pH irritation. Now, we have mentioned skeletal muscles, and that's a good place to end. On Thursday, I'm going to commence our lecture by talking about, very briefly, about cardiac muscles. So what I'm going to do is stop the slideshow, and I'm going to stop the recording. If this works, the recording